Hi students, now we'll move to the fifth class of animal viruses, which are double-stranded RNA viruses. This viruses of this category has got segmented genome. The double-stranded RNA is segmented and each chromosome which encodes a single polypeptide. The double-stranded RNA act as a template and synthesize a positive stranded or plus stranded mRNA. That is, this is double stranded RNA. This double stranded RNA that synthesizes the mRNA. And another feature I already told you, they have got segmented genome. Each genome produces its own mRNA. Examples of this category are reoviruses. Example is reoviruses causing infection in human beings. Now we will move to the sixth category, which is a special group which has got a single-stranded RNA genome. They are single-stranded RNA viruses which has got a plus stranded single-stranded RNA viruses, but they have the feature that they carry the enzyme reverse transcriptase. This reverse transcriptase, they can synthesize DNA from RNA. They can synthesize DNA by using RNA template. So how these viruses multiply is that this single-stranded RNA viruses that synthesizes a negative-stranded DNA. A single-stranded DNA will be synthesized from single-stranded RNA using the reverse transcriptase enzyme, and that act as a template for the transcription of mRNA. That is, here the plus-stranded RNA is converted into minus-stranded DNA, which act as a template for the mRNA. That is RNA into DNA and then to mRNA. This occurs with the help of reverse transcriptase enzyme. Examples of this category include retroviruses. Retroviruses, example you have heard about the HIV viruses and Rous sarcoma virus, mouse leukemia viruses. Retroviruses are of different types. So this is a special category with a reverse transcriptase enzyme. They have got a reverse transcription mechanism. Now we will move to the seventh class or the last class of Baltimore classification system, which is also special, which has got a double-stranded DNA with reverse transcription activity. They also carries reverse transcriptase enzyme, and you may think that how these viruses use why these viruses use reverse transcriptase enzyme, because they will carry out their activities in a special way, because this double-stranded DNA of this virus is actually segmented, or it is not complete, and there is a gapped genome. This is a double stranded, we can call it as a gapped genome. That is, in between certain segments we can see certain gaps, and these gaps are filled by this RNA. That is RNA, this RNA serves as a template for viral reverse transcriptase enzyme for the production of a double stranded DNA genome. That is, it will form a closed circular DNA with the help of this reverse transcriptase enzyme, because the RNA produced by the DNA act as a template for this. For this, they use the reverse transcriptase enzyme. I think it is clear for you, because these viruses carry a gapped genome. For the completion of this gapped genome, they synthesize DNA by using the RNA template, and this RNA template is converted into DNA using reverse transcriptase enzyme. These examples of this category are hepatitis viruses. That is a Hepatinaviridea family. Example is hepatitis viruses. So this is the last type of or last class of double-stranded DNA, last class of animal viruses based on Baltimore system of classification. So we'll see an overview of the different categories of viruses. That is the double-stranded DNA viruses we have discussed, and they have got different types of virions, and also the examples I already told you: adeno, herpes, papilla, pox virus, etc. And the single stranded DNA viruses are the, and then plus stranded RNA viruses are the, plus stranded single stranded RNA virus. Here plus stranded means this RNA itself acts as a template, itself acts as a mRNA. That RNA of the virus act simply as mRNA. Whereas the minor strand RNA, that is, it produces an mRNA from that. Whereas single stranded RNA viruses are the, and then also this plus stranded single stranded reverse transcriptase enzyme is the, and then double stranded DNA viruses with reverse transcriptase activity is the. So in this topic we have discussed a brief introduction to, I'll briefly about the structure of viruses. We'll see the detailed structure and how this animal viruses multiplies in this host cell in the next session. And we have discussed about the classification of viruses, the different criterion for classification of viruses, and the most important classification system of viruses, that is Baltimore system of classification. And based on the Baltimore system of classification is based on how this viruses uses the genome for the production of mRNA and protein to produce the infectious virions. And based on this, we can classify viruses into 7 classes. And we have seen certain examples of these classes also. Now, okay, I think it's clear for you. Thank you.